Welcome everybody to 52 Living Ideas. Today's Comprehensivist Wednesdays that we do in collaboration with the Greater Philadelphia Thinking Society. It's always delightful to have Sanjay. He's been doing this incredible series on neuroscience. If you have missed any of these YouTube videos or meetups, please go ahead in the 52 Living Ideas YouTube channel and look for Neuroscience playlist, where you will find all of Sanjay's meetups. So Sanjay, what are we discussing today?
Today we have a interesting topic. One, I always try to mix in contemporary and even philosophical or ideas that can energize and engage people, along with the information around neuroscience. So what I'm going to do is the first part we're going to look at. Basically, I'm going to set up the second part, set up in the sense that to understand the second part, we really need to understand a little bit more about what's happening in the brain, and especially with neurons and neuronal circuits. So I'll do that in the beginning, and then we'll dive into the issue of free will and decision making, and how that there are two particular studies that I'm going to focus on. They are similar, but the second is much more robust, and it was more recent. But they really give us a quandary in really, in terms of whether free will exists. A lot of the findings are they're controversial because other interdisciplinary areas of knowledge, especially philosophy and even physicists, have some disagreement with the findings, but anyone who's looked at the long breadth of research around this specific area will find it very difficult to disprove or dispel this. So we'll go into some of this tonight.
Excellent. Let's proceed.
Okay. So let me put the slide. Let me just do a screen share. So what I want to do is first, hopefully this is the right one. I think, yeah, so I think everybody should be seeing this opening slide. So basically what I'll do is first I'll go into decisions, and not simply decisions, but specifically, a little further down in a few slides later, we'll start going into exactly what's happening inside the brain when we're talking about decisions. But first we'll talk in general about, decisions and what they are and what they're about. So the first thing to understand about any decision, and this is not, I'm not talking only about humans. I'm talking about any kind of animal with some type of simple brain that is able to make, what we call a decision. Now, a decision at its simplest level is simply an action. We could think of it as an output or a result, something that happens that must be initiated. That is in my mind, the simplest that you can get at. Basically, there has to be an initiating event or action or some type of stimulus, signal, for example, and that causes an action. And the initiation of the first act is actually what causes the second act to follow. So this has to, and just in terms of the way that we look at this, I'm going to explain this in terms of this occurs within implicit systems. So a system is basically a constrained space within a constrained time, within which these actions and initiation, etc., occur. So, you know what? There's a. I thought I'd gone through this, but I missed this one slide. Anyway, let me just continue through, and so there's supposed to be a box around this. So basically, what I'm trying to show is that the system consists of a box. There's supposed to be a rectangle, and what's inside and what's outside. These are the two main elements of a system. And in the simple example that we have, well, this is really not working well. Anyway, so in the simple example that we have, in this system, we have this planet Earth, and there's a little house on the planet Earth. And in theory, what should happen is when we have, actually, this is not working at all. So anyway, I'll talk through this. Sorry about that. I thought I had tested these out beforehand, but so I had copied the file from another computer, so I should have tested it on this computer. So basically what I'm trying to show here is that in this example, I had like, many of you may know of this thing called the clapper, which we had, years ago, more than 2 decades ago. It was a little device that you installed around a lamp in your house. And when you made a clapping sound, it either turned on that lamp or turned off that lamp. And the concept is that you have to make the clapping sound near that device. If you make it far away, for example, in outer space, far away from your house or that device, it's not going to have an effect. So the system is something that is very important in the sense that the input that you have, for example, the clapping sound that you make to energize this device, it has to be within the system. It has to be near the action, the activity that's going to cause the action that's going to cause. So there's an input in any kind of decision. Second, there's a result that comes out of the decision, which is the output. So in this case, it would be the light turning on or light turning off. That would be the output. So the input would be a person clapping their hands. That actually the sound and it can be other types of sound. You can even have a recording, an audio of a clapping of a hand, and the system doesn't know if it's actual hand clapping or if it's just a recording. It simply bases it off of the volume and the type of audio that's coming out. Now, the input and the output have to be measurable or observable in some sense. And in the physical world, most everything that we deal with are either observable or measurable in a sense. So, most decisions can be, most activities can be thought of as decisions. Again, the input and the output both have to be within the system. And these are just very basic requirements for any kind of decision that happens. The saying that the will finds a way, that's basically a simple way of saying that basically the will is, in our mind, it is the initiating act. It is the initiating signal, and usually it's information, but in the simplest sense, it is a signal. And that signal then, throughout our brain, it may move to one area or it may move to multiple regions, and that will cause something else to happen. So if we have a will to raise our hand, there has to be a signal in our mind that occurs at some point and in specific places. And specifically when we're talking about raising our hand, it has to go into the motor cortex, the edge of the frontal lobe, and that signal has to then propagate, and then it has to move to the specific area where, if you're moving your right hand, it has to go to the specific area where the muscles that allow you to move your right hand, they get energized from that initiating signal. And then the output is, you can think of the output as both the neuronal signal that gets sent to the muscles in your arm that actually causes the arm to rise, or you can think the actual rising of your arm, that may be the output. And in fact, both are tied together. And in that sense, you can subdivide a decision into multiple decisions. So decisions typically in the real world have cascades. We don't just have one decision. For example, if you make a decision to buy a car, you also have to make a sub-decision on what model car, what make, what size, etc. Even the color of the car. These are subsequent decisions you have to make this overall decision of buying a car. So simple version of what a decision is. Now, decisions basically, at the top of this, I use the term signal or information. And signal and information, anyone who's, in these talks, we've talked about this to some extent, but anyone who understands this is not simply a concept from physics, but in philosophy and many of the sciences, many of the broad areas, not just the sciences. Information is something, and information also transfers into energy, but basically a signal is information. That's important to understand. Now, decisions are an aspect of causality, that one thing leads to or causes another thing. That's very important. What that means is that there's a cause and effect. And another way to talk about cause and effect is input and output. In the previous slide, we talked about input and output. But basically, the cause or the input is what initiates. It's the information or the signal that initiates. So, for example, our will to do something, that will is inside our mind, inside our brain, it's in the form of a signal, of a neuronal signal. It's actually spikes, multiple spikes, but it's not just a constant signal like we have in other things that we're familiar with. It's very different in the brain, but still it's a signal. And that signal is what, in our mind, when we think about it, it's what we do. We create that signal when we want to take an action, when we want something to happen, when we when we want to cause something. But we have to generate a signal inside our mind at that point. And then that signal, when it propagates through and various things happen inside our head, it eventually reaches to a part of our brain, which will cause that thing to happen. Now, that thing to happen might be actual motion through our body. It might be another feeling to come out. It might be, our vocal cords to enunciate and sound to come out, so we speak something. It can be something like, it may change our mood. So, for example, we may actually, it may, for example, if we're exercising, it may actually be something where we change our rate of breathing so that it calms us down. So it can be any type of thing which either affects our body or it affects the external world outside of our body. It can be, the effect can be internal. So it can be internal to the mind, it can be internal to our body, or it can be external in the world that we live in our environment. And the key point here to understand is that we're talking about flows of signals. So signal is a very important part of this. And then, next I'm going to go into, this is going to go a little more into the neuroscience of it, especially the network side of it, because signals are really what we're talking about when we're talking about decisions, that there are information encoded as signals in our brain that starts from one location where, our will originates from, if it is one place, but even if it's in multiple places, all of those places together have a concomitant set of signals that work together to drive the effect or the output of our will. So signals are a very important part of this. Here I have a very simple image, and this is actually, it's not a photograph of a neuron, but it's a replicated, it's a reconstructed image. So it is a realistic representation of what a real neuron is. This was a real neuron, and what this shows is, that this large bulb in the front is the soma, or it's the part that contains the nucleus. It's the part that does the actual activity of the neuron. And a little bit to the left and lower than that, these are basically dendrites. And this neuron is a specific type of neuron, so it doesn't have many dendrites. Other neurons have a huge a tree of dendrites. And then proceeding upward, outward from it is this long, progressively thinner and thinner area, almost like a spike. And that's known as the axon. And that's actually what carries the signal out of this neuron. And in theory, what it does is it carries a signal, and then subsequent to that, in the latter, toward the end of that axon, is another neuron. Actually, the dendrites, these filaments of another neuron would be around there. Now, actually, you can have a neuron here also in the middle, and those dendrites, because you can see these spurs emanating from the axon. Neurons can be very, very complicated in shape, and there are many types of neurons. So just to give an example, basically the neuron sends a signal down its axon, and eventually, either midpoint or somewhere along its path, that signal gets transferred. And if there is another neuron nearby, it will excite that other neuron. And the other neuron is not touching. So neurons never touch each other. That's important to understand. There's actually an interstellar space between them. And neurotransmitters, neurochemicals, there are various types of molecules which are emitted from the presynaptic neuron, and they go through this synaptic space, which is it's not a vacuum, it's a intercellular space, but it travels through that space through a finite amount of time. It's much slower than the neuron signals, electrical signals that travel, and then it enters the postsynaptic part of the next neuron. And so this way, signals propagate from neuron to neuron. Now, the next slide, I'm going to go a little more schematic into this. So this is a picture of basically the same type of neuron. Here we have the cell body, or the soma, at the top. And we have the dendrites at, above that. And then this long axon. And then toward the end, we have these, this is nerve ending is kind of a generic term, but basically these are post-axonal presynaptic extensions, and then there would be another synapse. There would be another neuron here. And what's important here to understand is that the signal or the information only and always flows in one direction. In every neuron, it always flows in one direction. It starts from the dendrites, it goes into the cell body, and once the cell body, does something, and in a simple sense, we might call it a decision, but it's actually not a decision. A decision we'll get to a little in a few slides, but it basically activates, it gets energized, and then that signal comes out of the cell body through this axon to a determinus down here. And then if there's another neuron down here, it will activate that other neuron. Or if there's something else here, for example, we may have sweat glands, or we may have a muscle tissue. So whatever it is down here, that those other types of tissues will get a signal sent to them to activate them. Now, in this example, I'm going to show basically two neurons. So let's say that we have a neuron back to back. In this case, it's a little easier. So the information flow still is going to be one directional. Even when the information comes back into the second neuron, it never will fly back. Will never flow back into the second neuron. It will always flow down through its axon to the end, and then if there's anything else there, it will continue down that chain. So if you see these two neurons, you can continue this chain down through thousands and millions of neurons and have this long chain of neurons. So in the simplest sense, you can have neuron chains which basically propagate a signal further and further and further downstream. But in the real world, in real brains, neurons are connected not just in this simpler linear fashion, they're connected in much more complex ways. And a few slides down, we'll go into that. I'll explain that in a little bit. Another point I want to bring up here is that these are actually not that different from transistors, believe it or not. And it's not identical, but there are, there are enough similarities between the way transistors work and the way neurons work that we can actually use this as an analogy. And this is actually one of the reasons why we've seen that artificial intelligence, computer-based artificial intelligence, seems to show a lot of the promise that our brains do. And there's a lot of similarity in terms of complex behavior that they're able to engender. And basically, a computer is simply billions and sometimes trillions of transistors. That's all they are. It's just as our brain is, 100 billion neurons. So in that sense, there's a lot of strong parallel between these. They're not identical. A transistor has basically three parts. A neuron has two primary parts. That's different, but you can wire a transistor to act identical to a neuron, and you can connect neurons together, two neurons together to act the way a transistor does. So there are parallels, and one can be made to work exactly the same as the other. And again, this is the reason why in other fields we are able to simulate brain-like behavior to such extent. Now, the next few slides, what I'm going to do is I'm going to go a little more into, now, those of you who may have attended some of the prior discussions on this, I've gone into some of these, but I need to go back just in case new folks are not familiar with this. And I have some, the prior discussions we had actually goes in much more detail of this. I have just a few snippets of this, so this is a little abbreviated, but I want to, give a little bit of understanding of what actually happens. It's not that the signals that flow through a neuron are like a signal that goes through the wires in our house or electricity. They're not the same way. So in this animation, you're going to see this on the upper left side. There's a battery symbol, and there's a switch. The switch is this little red arrow, and this blue circular thing in the middle is the neuron. And there's these two black probes, which are basically, they're probing the input. So this left side black probe is going to show the signal that is sent into the neuron through the dendrites, you might say. And the second probe is showing what will come out of the neuron, you might say out of its axon, the signal that comes out of its axon that gets passed on to the subsequent neuron. And this is actually something that's been done experimentally, tens of thousands of times by many researchers. This is in neuroscience graduate courses. This is something that most students do in the lab to actually get a feel for exactly what happens. And this is easy to simulate. So what we're going to do here is we're going to have this switch make a connection so that energy flows into the neuron. And then we're going to see here, and you're going to see two graphs here. One graph is going to show the energy flowing in, and the other graph is going to show what type of energy flows out of the neuron through the axon. And that's what I want everyone to understand here. So I'm going to turn this on, and you're going to see, again, this is one, the red is the input, the signal. So this point is when the switch was turned on. So the switch is kept static on, and then the switch is turned off. Now, the output that comes out of the neuron when this happens is it's not a simple type of signal that comes out of the neuron. What this neuron actually does is it has spikes. Okay, so in this case, we see 6 different spikes. So the number of spikes depends on many, many variables. But in this simple model, in this simple cartoon image, the duration that the switch was left on for, and the environment that the neuron exists in. So, for example, the calcium, potassium, other types of ions, and the proliferation of channels that are in the cell body and the axon, etc. Many biochemical effects that determine exactly how many of these spikes there will be, how large, the amplitude, the strength of these spikes, and the frequency of them. All of these factors are dependent on the biochemistry of this. But in a simple sense, while the switch is on, what's important to understand is while the switch is on, we're not getting a constant output. What the neuron does is it sends a pulse, then another pulse, and another pulse, and another pulse. It keeps sending pulses over and over and over while it's getting an input. Now, if you think about what this means, then in this example, we have a simple switch that's giving a signal in. Okay? In the real world, the neuron that would precede this would not be this way. It would send a single pulse in. So you would have a single pulse coming in. That single pulse it might or might not, depending on the amplitude and the type of signal it is, it might or might not activate this second neuron and cause it to send an output. But what you'll see is that the output from the first neuron will cause the second neuron to fire only once. But if this first neuron has a very, very strong signal, then this second neuron might actually have a pulse train. This is known as a pulse train. It might actually send multiple signals out. So it's a little complicated. We're not going to go too much into this further, but I want everyone to understand at a basic level that we're not talking, it's not simple, that it's much more complicated. And so the way the network is set up, the way these neurons connect together becomes very, very important. And I'll get to that in I think 2 more slides, or 3 slides maybe. So anyway, so let me go. So in this slide, what I'm going to do is I'm going to show that, so that what we have here is 3 different neurons, the green, the red, and the blue. There's 3 different neurons. And here, the green neuron is what's known as an excitatory neuron. The red neuron is what's known as an inhibitory neuron. And the blue neuron is the one that both of these are acting upon. And these words might give you a hint as to what's actually happening. The green neuron, what it does is when it fires, it excites, or it raises the possibility that the blue neuron is also going to have an output, that it's going to be excited and it outputs a signal. The red neuron, the inhibitory neuron, does the opposite. When it fires, when it sends a signal, it actually suppresses activity in the blue neuron. So if the green and the red neurons both are firing, then because there's excitation and inhibition, the blue neuron will not fire. Depending on the timing and the strength of the green and the red signal, okay, the blue neuron will fire a little bit, it will fire a lot, or it will not fire. And when I start this animation, this again is an animation, when I start the animation, there are 3 different combinations of this that it'll show, and I'll try to explain that a little bit, because this is important to understand. So first, we're going to have, let me pause it at this point. Actually, that didn't pause. Let me see if I can pause it just here. Okay, so first what's happening is the green neuron is firing just a single pulse. Okay, it fires a single pulse, right? And up here, so basically you have 3 different graphs here. Okay, the red is showing the red neuron, the green in the middle is the green neuron, and the blue on top is the blue neuron. Now, what's important to understand about the blue neuron is there's this dashed line that is a threshold, and that's important to understand because that threshold is kind of tied into the biochemistry of this system, and that will determine whether the blue neuron sends an output or not. In this case, over here, you see the spike. So And I'll explain that. So the first signal that the green neuron sends causes a signal inside the blue neuron to rise, but it rises up, but it does not go above this threshold. And then it normally cascades down and then becomes quiescent again. It gets back to the baseline level. So when this green neuron sends a single pulse, it doesn't do much for the blue neuron. It causes it to get excited a little bit, but the blue neuron doesn't send an output. Okay. The second time, when the green neuron sends two pulses back to back, and what's important here is that these two pulses are close to each other. So the first pulse, what it does is it causes the blue neuron to go up a little bit. And while it's still high, the blue signal is still high, the second pulse from the green neuron comes, which raises it even higher. Okay? And that blue neuron signal then, or the activation within it, gets high enough that it crosses the threshold, and instantly there's an amplification effect. I won't go into the detail, but basically this neuron then gets enough energy, it has enough energy, it's built up enough energy that it can actually shoot out, it can emit a very strong pulse along its axon further downstream. And that's what we see here in this example. Okay, so this is the first example where just the green neuron is acting. The second example here, as this continues further, we're going to see just a red neuron act. Sorry, a little bit of a difficulty with getting the animation. So the first neuron activates, then the red neuron is going to activate here, and let me pause it. So here we have the inhibitory neuron. Okay, and again, remember that this, what it does is it causes the blue neuron signal to go down, meaning it's harder for it to, for this, you can see this blue signal is going negative, it's going downward, which means it's further away from the threshold, so it's harder for it to go above the threshold for it to emit a signal. So the red neuron is doing that. It basically causes the signal to get less so that it's harder for the blue neuron to fire. Now, if we continue this animation further, we're going to have both of these, the blue and the red and the green acting together. In this case, what you see, the red neuron fires first and it causes the blue signal to go down, okay, to a fairly negative level. And then we see two green signals, the green neuron sending two bursts, rapid succession, one after the other, which rises it pretty substantially. But because it starts off with the red signal, the red neuron causing suppression, even the two green signals together, originally, if you remember, two green signals were sufficient to cause it to excite. But in this case, because the red signal preceded it, those two green signals are insufficient to cause the blue neuron to fire. And this is important to understand because these three neurons together are acting as, you might say, the simplest decision-making system in our brain. And I'll get into that a little bit further. And what happens there is that, there's no activity, at the end of that. This is just going to play through again just to show that that's actually what happens. There's no signal at all at the end. So in the next slide, what I want to do is, and this is what I said, just that these two neurons, and at a minimum, you need two neurons that come together to make a useful decision. In that prior example, we had three neurons. The blue neuron was the one that made the final decision. And you might say that the red and the green neurons were neurons that were acting to initiate, another neuron that was acting to, one was a will, and the other neuron, you might say, was a won't. Okay, so in our mind, when we think about will, free will, there's actually a complement to free will, which, in the research side is written or described as a won't. So there's a will and there's a won't. And that green neuron was a will for action to take place, and the red neuron was the won't to suppress the action to take place. And in people, when we want to make a decision, we also can choose not to make a decision, to withhold a decision. So these are examples. So in that case, there were three neurons, but at a minimum, you need two, one that actually causes the excitation and the other one that actually causes the decision to happen. Now, it's important that they have to be connected in the correct way. That's important. And also, there are many different types of neurons. I haven't gone into this here, but because of the many different types of neurons, the connections have to be done in a certain specific way. I'm going to go into a little bit, two slides down, but these are aspects that are very, very important for this. Now, there's another example that I have here that's going to show this, and this is going more into a real-world example. This is an example where, if you've ever been at a doctor's office and the doctor uses a little, hammer, a pinpoint hammer. It's not really a hammer, but it's a rubber mallet to hit your knee. And the doctor's trying to test your reflexes. And the neuronal circuit that allows this to occur is extremely simple, and it doesn't even go to the brain. It stays within our spinal cord. This is one of the reasons why this is a test for, this is not a test of brain activity. This is a test of simple, our central nervous system functioning, our spinal cord functioning, and our musculature functioning. It's a much simpler test because it doesn't require the brain to be active. But the circuit is extremely simple, and in the simplest sense, you could think of it as 3 neurons. Okay, and again, in that prior slide that we had, where we had a green neuron, a red neuron, and a blue neuron, this is the exact same analogy. Those 3 neurons are basically the same type of neurons that exist within our body, but they exist. All 3 of those neurons exist within our spinal cord. Actually, 2 of those neurons exist within our spinal cord. The third one exists. Sometimes it exists within our muscles, or within our patella, where the knee is, in that region. Sometimes it exists within the spinal cord. Now, let me just elaborate a little bit. It's not so simple to say that there are only 3 neurons. The real circuitry that is involved here is in parallel. So these 3 neurons, there are multiple copies of these 3 neurons, multiple, several hundred, maybe even thousands of copies of each of these 3 neurons. And that's what allows the signal strength that actually causes a muscle to activate to be that strong. It's because a single neuron would not be able to activate an entire large muscle in our leg to move it. You need a lot more signals from multiple neurons. And also this interneuron, this black neuron that you see, that was not in our original diagram, but this exists. This actually also does other things, and it causes amplification. And the real complex circuit that exists in our body, this red inhibitory neuron, there are actually multiple other types of inhibitory neurons here that go into the circuit. For example, there are multiple excitatory neurons. So for example, we also have an excitatory neuron coming from our brain. So for example, this muscle that we are able to flex, this muscle we can control from our brain. So there is a signal that comes down all the way through our spinal cord, down to this particular disc. And then there will be another green neuron that connects into the interneuron, that connects into this blue neuron that can be used to activate our muscle. So when the signal comes from our brain, it's a green neuron, connects into the blue neuron, and it causes it to activate. Excuse me, that's how our muscle normally is controlled from our brain. But one thing, if you've been in this situation, what you'll understand is that even if when the doctor hits your knee and your muscle, the reflex action occurs, even if you try to force your muscles to contract and to prevent that reaction, you're unable to. It's impossible to stop your body from doing it because of these inhibitory neurons. Because the system has these inhibitory neurons, what it does is it inhibits the signal from our brain from acting in this case. And only the signal from the hammer, the neuronal sensing, there's basically skin sensors and cell sensors, muscles that detect the vibration and the force, from the hammer. Those sensory neurons are the only ones that can cause activation, and because the muscle that allows you to prevent the motion is inhibited through this red neuron, it's impossible for you to stop this reflex from occurring. Even if you think about it, no matter how strongly you think or how strongly you exert energy to do it, you cannot stop it. This is why this is a reliable test of our spinal circuit. So just a quick animation, nothing complicated. Everyone knows what's going to happen. So the green neuron sends a signal, and it goes through to the blue neuron, and it causes the muscle to contract and causes the leg to move. But at the same time, what you're going to see is this green neuron sends the signal, and this signal actually splits into the red neuron. You're going to see here, there's a activation of the red neuron. You see a white dot here that appears for about 2 seconds. And that activation stays active and keeps this flexor muscle taut and energized so that it is not able to, it keeps it inactivated so that it's not able to be energized. And the extensor muscle is activated and energized, which causes the leg to actually move. So this is a very simple circuit, 3 neurons again, if we ignore the interneuron, but maximum 4 neurons. But again, this set of neurons are in parallel. So it's not simply 3, but this simple circuit, this microcircuit of 4 neurons is duplicated over and over and over and over several thousand times within our spinal area, which allows this to happen. But basically, the circuit is as simple as these 3 or 4 neurons. That's the minimum that you need to cause this type of action. Now, some of you might say this is not a decision, but let's go into that a little bit. So just finishing this. So these 2 neurons are working together to make this quote unquote useful decision. So here the decision is initiated by the doctor. Okay, it's initiated, but the decision is not made by the doctor. The decision is actually made, meaning the action that occurs by the 3 neurons working together. Okay, and the way to look at it is the decision that the neurons are making is, do we make the muscle move? Okay, reflexively. So the neurons are the ones who are deciding which muscle to move, which muscle to prevent from moving. So the motion that's occurring in our body is entirely controlled by the neurons. For example, the fact that the inhibitory neuron is there to prohibit movement, that's actually part of the decision. This is a complex decision in that it's not simply making a decision to move one muscle, it's making a decision to move one muscle while suppressing movement in a second muscle, because both these muscles work together in our body to allow certain movement. So this is, and this again, this simple circuit evolved through evolution through hundreds of millions of years, and this same circuit exists in pretty much any kind of moving animal that you see that has a very simple leg. And this is the most primitive type of decision-making system you can think of that allows us to do this. Now, let me go to another type of system that's a little bit simpler, even more simpler. This is the light switch that we all are familiar with. And the light switch basically turns the light bulb on or off. Okay, when the light switch is off, light bulb is off. When the light switch is on, the light bulb turns on. Okay, and in that sense, this is similar to, but in this sense, there's no not necessarily a neuron, but the wiring of the circuit. Okay, the wiring of the circuit is what's causing this to happen, in the sense that the switch has a internal to it. It has contact that makes and breaks. And the wiring is extremely simple in that, and these you can think of as axons and dendrites. And this light bulb is the terminal neuron that sends a signal. That signal happens to be light, photons that it emits. So this is the simplest circuit that you can have, and this is similar to the first diagram that I showed, where you had a simple switch that activated a single neuron. This is basically that type of circuit. The next type of circuit, this is a little more complicated, but this is more real world, and this is more getting into, in our houses, we have these circuits, but these circuits are a little more complicated. So in a smart home, you might have a circuit that is more like this, where you have a light switch, but the light switch isn't the only thing that controls whether the light turns on or off. You may actually have a motion sensor tied into this, and I haven't drawn this in a complex way because the actual circuit would be much more complicated. I don't want to make it messy and difficult to understand, but the point is, all 4 of these things together, this switch, this box, and this box, these 4 things work together to decide whether the light bulb turns on or off at any given moment. So for example, there may be a motion sensor, which determines if a person walks into the room, then it turns on based on, you being in the room. And when you leave the room, there may be, most motion sensor people know about this, there's a timer built into this. So after a certain number of minutes, 5 minutes or 20 minutes, it times out and it turns the light off for you because you've left the room. So this is one type of complex decision-making that's built in, and this motion sensor itself will have its own set of, you might say, neurons or transistors or a circuit. It will have a circuitry inside of here that will do the complex aspect of that. And then there might be another module, you might say, in this circuit, which decides is it daylight savings time or not. So exactly, depending on if it's nighttime or daytime, and if daylight savings time is on or not, it will determine whether the light turns on. For example, this might decide that if it's daylight on this particular date, then the light will not turn on. So this will only turn the light on when it's dark or evening, and when daylight savings time is appropriately met. And again, this would be a more complicated type of circuitry that does all of that. And then there might be another circuit that basically figures out and decides or knows when the homeowner is home or not. So if the person who's living in the house, in this building, in the house, if they're home or not. And if no one is home, then even if these other things, maybe you have a cat, and if the cat moves and it's nighttime, well, this might decide, well, the cat is not good enough. It's not going to turn the light on for the cat. You might, again, this is a programmable system. So in your house, you might actually set it up so it does turn on for your cat. But the point here is that this is a complex decision-making circuit. And similar things like this exist within our brain, except these exist in different parts of our brain. And many times when we're making a complex decision, this is actually what's happening is, part of our parietal lobe, and part of our temporal lobe, they both are involved in aspects of a decision. When you will a decision to occur, those other parts of our brain also provide input into the overall decision, whether it will happen or not. For example, if you're I'm trying to think of an example. if you're a child and you're playing jump rope with your friends, well, when you're playing jump rope, your body is trying to make sure that you don't, because you're jumping up and down, and you have a specific rate at which you're jumping. So you have to make sure that you jump based on what your eye is seeing, the rope moving under your feet. And you have to time the movement of the rope. And if you jump at the wrong time, that rope might hit your legs and cause you to fall and trip and get hurt. Another aspect is, as you're jumping, your body is keeping its own balance. The proprioception is very important, so that you don't, if you jump in a certain way, your body needs to maintain its balance. So that's another aspect of your brain that is active. That's another one of these modules which is active, the cerebellum in that case. So there's multiple parts of our brain that are always doing things, even things that appear to be very simple, like jumping rope, skipping rope. There are actually many parts of our brain that are actively involved to keep us safe and being able to function in that realm. The next slide, this is where I'm not going to go into detail in any of these, and there are many, many other things, but this is basically a slide that shows some of these neuronal circuits that I talked about, micro networks that I talked about earlier, that these neurons have to be connected together in certain ways. And depending on how they're connected together, you can get very complex behavior. And these are just 8 examples. And again, the greens are the excitatory neurons, the reds are inhibitory neurons. And as you can see, some of these, this excitatory neuron actually activates both of these inhibitory neurons, and signal goes outward to whatever it's connected to. So this is a fairly complicated type of circuit. Here we have a very, very complicated circuit in the bottom left, where you have these 1, 2, 3, 4, 5, 6, 7 excitatory neurons, which cause 3 outputs to occur. And in this, and there are 3 inputs to occur. In this case, it would be fairly complicated. Here in the middle bottom here, you have 4 inhibitory neurons, and what this actually does is it causes like a pulsing action where only one neuron will turn on, and then the next neuron will turn on, and the next turn. So this will be almost like a light, 4 different lights. One light flashes on, then it turns off. The next light flashes on. So the behavior this will cause will be similar to 4 different lights where only one is on at any given time. So you can get very complex behavior just from a few different neurons. That's what I want people to understand. And again, you might think that these are not decisions, but think of it this way: that if this neuron, or not here, let's take an example of this up here at the top, where you have 3 green neurons and two red neurons. So out of these 3 green neurons, if these 3 green neurons are, let's say, the parts which are about jumping rope, okay, so one green neuron affects your balance. That it basically culminates the signal that comes out of your entire cerebellum that gives you a feeling of being in a balanced state or not. Either you're balanced or you're imbalanced. Okay, so this one green neuron will give you an output that helps you establish whether you're in a state of balance or not, physical balance or not, meaning you're about to fall over or not. Another green neuron here might come out of your visual system, and it basically determines at this instant, is the rope going to hit my leg or not? If the rope is not going to hit my leg, that means it's safe, right? If it's going to hit your leg, then that can get you into trouble, because if you're jumping rope and the rope hits your leg, the rope is going to maybe tangle your leg. It may throw you off balance. It may cause a problem. And this middle green neuron might be a timing neuron that you have within your brain, because when you're jumping rope, you're keeping track of time. And so that may be a rhythmic neuron or set of neurons. So again, each of these neurons represent entire regions of brain activity, complex behavior in and of themselves, which send an output, which is a single signal So each of these 3 green neurons represent the output of complex circuits that goes out, and then they terminate into 3 different signals, go into this microcircuit, and then similarly, the red neurons might be inhibitory, which might, for example, there may be another inhibitory signal which is using your occipital, which is using your vision system. And if your vision system is realizing that the rope is too close to your leg or the timing is off, it may excite, it may activate to turn off your impulse to jump at that moment. So basically, it stops you from jumping, because while you're midair in the middle of a jump, it's risky because you can fall over. You can fall and get hurt. So you don't want to jump when the rope is going to hit your leg. So this inhibitory neuron may be very complicated in the sense that it's using a visual system, and the output of all of that complicated analysis that your visual system does, even the peripheral visual system, even when you're jumping rope, you don't have to necessarily look at the rope. Your peripheral system is aware of all motion. It can see the motion of the rope even though you're not directly staring at the rope. So it is able to track the motion of the rope, and it may have an inhibitory signal to prevent you from jumping at the wrong time, which will cause you to fall. So again, this is a circuit that would help you make a decision of when do I jump or when do I not jump. Okay, so this is again a complex decision of jump or not jump. It's actually 2 decisions tied together, but this is an example of a circuit that would do that. So hopefully people are starting to understand that decisions aren't the way we think of them in the simple world, that you decide on something and it happens. There's actually complex things that work together inside our brain that can cancel a decision, or they can promote a decision or allow it to happen even faster or earlier. So these are all various types. And then let me pause here. So before that, what I want to do is I want to go into some points for a breakout discussion that we can have. So this is basically talking about decisions are propagated within and by neural circuits. And the question is, do you agree, or if you don't agree, because some of you may not agree with this, do you believe that decisions are propagated within neural circuits? And there are 3 parts to this, or 3 ideas around this, just to explain it. All decisions start as a signal from a neuronal cluster. Okay, so for example, you may have one of those clusters might be your cerebellum, which basically figures out whether you're in a state of safe balance or not. You're in a physical balanced state or not. And so these decision signals propagate through the brain to other areas. So it goes from a cerebellum and it propagates, and from what we understand of the brain, a lot of the more complex decisions actually occur, at least one of the areas that they occur in is the frontal lobe, more specifically the prefrontal cortex, but basically the frontal lobe. So those signals, many of the signals that we have that are will-based end up in our frontal lobe. So this is an important point, that all decisions start as a signal that propagates, that moves to other areas of the brain. Another part that's important, that's part of this whole idea, is that signal, that single signal, or you could have multiple signals. In some of the examples that I had, we had multiple signals going, but that signal can branch and become multitudinal, causing many resultant behaviors. So similarly, just as that signal of whether you're in a balanced state or not from your cerebellum will go to your frontal lobe to either tell you to jump or not jump, that balancing signal may also go to your left leg and your right leg, so that it helps coordinate whether the 2 legs jump, the muscles in both legs contract at the same exact moment or not. Because if you contract the muscles at different times, one leg will flex and the other leg won't flex, and you won't actually jump up, you actually jump leftward or rightward. It'll cause you to fall down. So that same signal may actually also go, not just to your frontal lobe, it may also go to your 2 legs to coordinate so that the 2 legs jump at the exact same instant. So these are important. These are just more complicated aspects of this neuronal circuit. This circuit is usually in the real world very, very complicated, involving millions and probably hundreds of millions of neurons. And the last point here, example is I gave the example earlier of jumping, skipping rope, as a child. Another example is if you have a memory of a childhood pet who died years ago. Well, that memory, and here, this example, it doesn't have to do with muscles. I chose this because this is an example where a memory can actually evoke a decision. So for example, the memory would evoke emotions, both an affectionate emotion, because if that pet happened to be very loving, very caring, a pet that really was very cuddling, then it'll bring out emotions in other parts of your brain, which have to do with affection. And it might also bring emotions in another part of your brain that has to do with fun, because it brings out other memories of fun. So it might bring out a feeling of affection and other memories of fun. So this single memory of that pet will generate other emotions and feelings, and those will propagate through, as the bullets at the top say, they'll propagate through and then eventually cause another decision. That decision might be that it brings out a memory of something that happened. Okay, and that's anyway. So that's the idea I want to leave us at this point with in the breakout room. If there's any questions, we can go into questions. But I want people to really think about, can circuits in the brain, or do circuits in the brain cause decisions? That's the fundamental idea that I'd like people to understand.
So, Sanjay, what I was thinking of is to go ahead and do the breakout rooms for maybe about 20 minutes, let people think about it in their own way. And then they come back and maybe do a quick round of questions at that point. So they get to think about their questions, discuss it among themselves, and get the best questions for you. And then we'll continue with the second part. Does that make sense?
Yeah, great.
Okay. So we'll do the breakout rooms for about 16 minutes or so. I'm going to have Allison, Maritza, Joe, and Sanjay facilitate the breakout rooms. Inside the breakout rooms, the rules remain the same. Just raise your hand if you want to speak. Keep on topic, be brief, and be courteous. And Sanjay has already given suggestions about what to talk in the breakout rooms, starting the breakout rooms now. Welcome back. All right. Welcome back. So, Sanjay, I was thinking, can you tell me how long your second presentation is? You're on mute.
Sorry. Yeah, second part is not that long. I can probably wrap it up in 15 minutes or okay. Around 20 minutes.
How about let's wrap up that, and then we can take all the questions together. Folks, keep track of all your questions that you already have, plus any new ones that come up. And then we will, as soon as Sanjay is done with the second presentation, I'm going to collect all the questions, and then we will try to answer all of them. All right. So let's proceed. Go ahead, Sanjay.
Okay. One thing though, before I continue into that, I do want to just present, because in the session, that breakout session that I was in, some very good questions, and there's some points that I think probably other people may have confusion or need elaboration on. Two things in particular. One is that I want to make sure people understand that when we're talking about signals and information in the brain, we're not talking only about information coming from the outside world. Okay, there's information within our brain. For example, memory is a type of information. Okay, and actually that memory eventually becomes a signal. So these microcircuits that I described are not dealing only with information from the outside. And this is what, once you understand this aspect of it, you start to understand the power of why this actually shows how our brain actually works and how decision making in our brain works, is that you can have multiple areas of our brain, memories, emotions, guttural, visceral feelings from your body, all of these things, as well as sensations from the outside world coming in. All of these things basically become signals within, they're not usually a single neuron, but they're basically large clusters of neurons, but eventually they coalesce into a small set or even a single neuron. So these complex signals, so for example, the feeling of hunger probably does coalesce into 1 or 5 or 10, it's a very small number of neurons, okay? And that will carry the signal of hunger, quote unquote. And similarly, when the description I gave with balance, right? If you're jumping rope, that feeling of whether you're in balance or not balanced, okay? You would have one signal that gives you this understanding of you are in a state of balance in this moment. And again, this is changing over time because as you're moving through space, as you're jumping, that signal in the neuron is changing constantly also. It's keeping track with whether you are in balance or not. So it's saying, yes, I'm in balance. Oh, not anymore, not anymore. Nope, now I'm balanced again. Like it's constantly changing. And then you may also have other neurons supporting it, which give you information about if you're out of balance. Are you shifted to the left? Are you shifted to the right? Are you shifted, twisted? You know, there's all these other neurons and regions of the brain, which all together give you a more complex understanding of not simply you're in balance or not, but how do you get back into balance if you're off balance, right? Which muscles do you have to move to get yourself back into balance? So all of this information is coming from, let's say one, in this case, it would be the cerebellum, one part of the brain. So there's information in the brain that's extremely, extremely detailed, but they coalesce into a small number of neurons. And then those small number of neurons carry this information into, let's say, if the will is exercised in a single area of the brain. We don't know that right now. We believe it's a small number, and the prefrontal cortex happens to be one of those that shows high promise. But if it's a small number, let's say 3 different regions of the brain that together work to give us will, right, or willpower, then those signals would go to one or all 3 of those regions, and then other signals would go to one or all 3. But there would be this microcircuit. And a microcircuit doesn't have to be localized to a small space. That microcircuit may be 30 different neurons. 10 of those neurons are in one region, 15 are in a different region, and 5 are in a third region. But those 30 neurons all together, split across 3 different regions of the brain, are acting as this microcircuit, which actually gives us the will, in this case, of jumping, of do we jump or not jump? So that's one idea that's important, is that these signals can arise from within our body and our brain, for example, memories. The second idea that's important is that let me try to remember. When we're talking about free will, and free will is, we've had many discussions in this group also about consciousness and the philosophical and the quantum mechanical aspects of it. There are many aspects to this. We're not going to go into much of that today. What I want people to understand today is that the circuits that we're talking about are, and again, you don't have to think of the circuits if you don't want to, but they basically distill down into very simple circuits. Although if you think about any real world situation where you're making a decision, that simple circuit would engulf probably parts of every aspect, every area of your brain. But all of those areas of your brain would send signals into these, finite areas which actually exercise the end will, the decision making. So these signals in these circuits are not about, like one person made a comment that they seem to be like electrical. They're not. The biological aspect of our brain works very, very similarly. Neurons, it's not the same, that the signal is not an electrical signal. They're rapid pulses, but our brain is an electrical organ. That's something that people have to understand. And so it's important to understand that this is not about, I'm not saying that our brain is like transistors. Okay? Even though I use the example of a transistor, that's representational, that the way information flows through a transistor and the way information flows through a microcircuit of neurons, there's no difference in those. It's just so that neurons are biological, purely biological, and it's more than just electrical signals. There are also neurotransmitters involved. So it isn't different from a transistor, but the behavior of signals and information flowing, that's the key part that I want people to understand. So if you have questions, hold for a few more minutes. Let me go back to the slides, and then we will go into Q&A. So let me just pull up the slide. Okay, so next, what I want to do is I want to talk about 2 specific experiments that are famous. In the write-up, I described Benjamin Libet's experiment, and there's a second experiment by Itzhak Fried, which is very similar, but it's much more elaborate and sophisticated, and actually gets to much more of the information. Now, I won't go into detail with Libet's experiment because Fried basically repeated many aspects of it are a repetition, but he added to it and he did much better work. Not that Benjamin Libet didn't do work. He did very good work, but the time in the 1980s, early 1980s, technology was not as sophisticated and it was limited in how far he could go. So his work basically started with a lot of research that started from the 1960s up into early 1980s. He started with that knowledge, and then he did an experiment that started in 80 and went on for 3 years. He had many subjects, multiple subjects, and he did with each subject, he repeated the test multiple times. So he collected a lot of data. And what he did is he asked each person was asked to decide when to press a button. And in a later slide, I'm going to go a little more detail with, there's a visual to help you to understand exactly what it was. And, but, this information applies to the Freed experiment as well. So, if you understand it, in Freed, same thing Libet did, but he did a simpler version of it. So each person was asked to decide when to press a button. For example, a button on a keyboard. Okay? Like you might press a space bar on a keyboard. The person was shown, and in fact, it was a computer that was in front of them, and the screen had certain information on it that the person was given, which helped them to make a decision on their own. So they were asked to press a button. And there was a lot of data that was collected as part of this experiment for each person. And the data collection was repeated over time. So one of the things that Libet did is he used basically their squid sensors that basically they collect through the scalp. So this was it was not invasive to any subject. He collected the signal. He had many sensors spread throughout the brain, the outside of the brain. He was collecting many signals from that person's brain as they were doing this activity, as they were pressing the button, as they were thinking, as they were deciding, when they decided to actually do or not do. All of these things were causing activity, electrical activity within their brain. And all of those signals of electrical activity were being measured by these sensors. They were picked up and then they were going and being recorded somehow. So that was important. And here and this was all done externally, so there's nothing inside the brain. Now, what's important here is that at Libet's time, this was the best technology we had. So the signals were weak and they were not specific, meaning you couldn't really tell exactly how many neurons it was. If it was a huge, let's say 1 million neurons that together were sending this signal, in reality, it would have been a large number of neurons because signals from a single neuron would not have been picked up by the sensors. They were not very sophisticated sensors back then. So that was one aspect that was a problem, but still he overcame it because he was able to collect signals. Second aspect of this experiment was that there were 2 clocks. Again, there was a computer in front of the subject, and the computer basically used these 2 clocks inside of it. One clock was visual, so it actually showed a dial moving on the screen. So that was a visual dial, which the subject was able to see, and the clock had numbers on it, so the person was able to basically make some decisions estimating when they actually wanted to press a button. And the second clock was built into the computer. It was not visible to the subject, but it was keeping track of all the signals that were being recorded and the button press, all of these things and the timing between them. So there was a lot of time information that was recorded here. That was important. Also, the data collected was exactly the moment of the finger press and the fact that the finger was pressed, because the person also had the option of not pressing the finger. In reality, every time they did press a finger, but if they chose not to, the experiment still would have continued and it would have found that the person did not press the finger. But that was an aspect. And then there was also an aspect of the data that was collected. This was the only subjective aspect. And that was that the person in their mind decided when they wanted to press the button. Okay. And there was no way for Libet to actually sense when that moment in time occurred inside their brain, when they decided, when they absolutely knew they were going to press the button. Okay. And part of the reason is because we don't know where decisions arise in our brain. We don't know where will exists in our brain. That was one huge aspect that prevented it. Even if we knew, the technology at the time was so crude that it would not have allowed him to go down to the level of a small number of neurons to measure that. He was measuring neurons, thousands, millions, and probably hundreds of millions of neurons at a time. So it was a very coarse type of measurement, but nonetheless. So in the experiment, what he did was he was asking, as this clock was rotating on the screen, the person was instructed ahead of time to look at the clock, and basically when they, in their mind, make a decision, keep track of where the second hand is on the clock, and it wasn't a slow moving, it was a fast moving second hand. So the precision with which they can tell when they chose, there was a good, good bit of detail in terms of being able to know exactly when they made their internal decision to press the button. So the person was asked, but they were asked based on what they were seeing on the screen of exactly when they pressed the button. And then the computer itself knew exactly when they did press the button, because that was physically connected to the computer. So all of these things together gave a lot of data to Libet, which subsequent to that he did an analysis on. Now, next slide, I'm going to go straight into Fried's experiment, because his experiment is very similar, but I'm going to highlight some important differences. And his experiment started in 2010 and finished in 2011. He built on all of the prior knowledge research, including Libet's work, and the experiment was very similar to Libet, but there were some key differences. He used 13 subjects, only 12 of them by the end were involved in the data, because one of the subjects, there were some issues. So data from 12 subjects were involved in the study. What's unique is that each of these subjects in Fried's experiment were unique in that they all had epilepsy, and they all had intractable epilepsy that was not treatable by medication. And so the only treatment at that time that they had was that they were implanted. They had surgery done separate from this experiment. The surgery had been done long before this experiment. But Fried found these people who already had these electrodes implanted in their brain. He found them, asked them if they wanted to participate in this subject. 13 of them said yes. And those 13 people, then he sat them in front of a computer. But because those electrodes had been implanted in these patients' brains already for other reasons, because of their epilepsy, and those electrodes were used for various reasons, we won't go into that. But basically, there were about 700 electrodes in each person that were implanted. So there are a lot of electrodes that were implanted in each person's brain. And these electrodes were implanted in a way that they were actually able to measure very, very precisely. In some cases, they were measuring down to a single neuron. Okay, many of the electrodes were measuring the output of a single neuron, which is the best you can get. And other electrodes were measuring very small clusters of neurons. And he described in the paper. That's important to understand. So he was measuring actual signal inside the brain and actual signal at single neurons, which is extremely important. The other aspects of this experiment were very similar to Libet's. Each person was asked to press a button. And basically the same type of data was collected, except the data around the signaling didn't come from external measurements. These were electrodes that already were in the person's brain. And those electrodes were allowing in the experiment to collect the exact moment in time, not just the moment in time, but also the state of the neuron throughout the entire experiment. So even when the person was doing nothing, their neurons were doing something. Their neurons were either sending signal in a certain state. So Fried was able to collect in real time every single type of signal that he could collect from all of these 700 electrodes, which were several hundred neurons for each person. And then he repeated this experiment over and over and over with each person. So he had, I forget the exact number, but there were quite a number of several hundred data sets he had of this experiment being run, which was more than Libet's. But the key point is that the data was around single neurons and small clusters of multiple neurons. That's a very important aspect. Other parts of this are identical to Libet's. He had two clocks built into it. One was visible to the subject. The other kept timing of all of these things. The subject was asked to press the finger, press a button using their finger. And then again, they were asked to subjectively rate at what time they made their decision to press the finger. But Fried also did, in this subjective part, he actually added an elaboration, which I'll get to in the next slide. So if you understand so far, the experiments are similar, but the type of data that Fried collected is much more intrusive. Luckily, he was able to find patients who were willing to participate in this. Unfortunately, those patients had another issue, issues, but this helped the neuroscientific community understand, these other aspects of their brain, of our brains, because those participants, agreed to help in this. and they were not harmed. There was no detrimental impact from this. So this next slide is where, and at the bottom I list the actual study if anybody wants to. It's a phenomenal study, but it does go into a lot of depth. And you know, if you aren't familiar with things that we've talked about in the past, some of it may not make sense. But the key part here is, and this is a representative slide that shows there are 4 specific regions where a lot of these electrodes were implanted, or at least the electrodes that he used in the study. The actual electrodes that were implanted in the subjects were more than just these 4. But these 4 regions were the ones that he focused on because he felt, you know, for various reasons, they were the most important. And also the density of electrodes that were implanted in these regions were high enough that he could actually collect a lot of data. So the SMA is the somatosensory motor cortex. The pre-SMA is part of the frontal lobe that is just prior to that, which actually acts to activate the motor center, which actually does the motion, you know, activating muscles. The accumbens and the dorsal and the rostral segment of I think it's the accumbens that the black and the green were also had electrodes implanted in them. So these 4 regions were where he was getting all of the data from. Now, something that is important to say here, and he elaborated on this in the study also, and anybody who does research, this is one of the things that you have to do in research, is you have to look at the weaknesses of your study design. You have to look at what possibly might be happening, what might not be happening, what it might explain, you know. So basically, because he's only looking at these 4 regions, even though he's looking in a little bit of depth, because he's only looking at these 4 regions, we can't say what's happening in all of the other regions of the brain and whether they contribute or not. So it might be that what he found in these 4 regions give us information, but it might be that there's another region that he didn't look at all, which has even stronger impact on the person's will. Okay, that might be possible. But Fried and Libet, like I said earlier, they had looked at prior research, decades of research prior to that. And those decades of research prior and a lot of the research subsequent to Fried have corroborated and supported this idea that the frontal lobe, most of these sensors are in the frontal area. In this study, there are sensors in the temporal area, in the parietal lobe. There are actually sensors in those other areas also that they collected. But in the summary and the conclusion of this study, he found that those did not, while they did show some indication, and because of all the other research that we have showing, the frontal lobe is the main area that seems to, you know, and not just this type of experiment, many dozens and hundreds of other types of experiments, not just around free will and consciousness, but around many types of mental functions, executive functions. All of those experiments have given the conclusion that these types of processes, mental processes, seem to originate or terminate in the frontal lobe. So this is the reason why the data that he elaborated on are these 4 regions, because they are parts of the frontal lobe. And even though he collected data in other parts of the region, and you know, all of that is available. Anybody who wants to reanalyze his data can do that. Most researchers who do things like this, they provide the data for other future researchers. They can go through everything with a fine-tooth comb and do better analysis if they feel they can do so. So everything's there. You know, it's open so that anybody can challenge and find fault if they feel there is a problem that his team had done. So this is one slide that shows that there were many, and what's also important is each of these dots actually is a region of, it would have been thousands, probably millions of cells. But what's important is each of these dots, this green dot, this black dot, etc., each point had 8 probes implanted in each of these dots. So what that means is that if this black dot here was, let's say it was targeting a single neuron, then there would be 8 different probes pointing around that one single neuron. So he was collecting a lot of data. So even if one of the probes, for whatever reason, wasn't getting a good signal, or the signal was distorted in some way, or there's some bias, all 8 of those probes chances are less that all 8 of those probes would have been affected by that signal bias. So the data he collected was very robust. It was very strong, and mathematically, and in terms of computational, in terms of a lot of the complexity around what he did. He had a lot of robustness built into it. And for example, in another region where he had another cluster, that cluster might not have been a single neuron, it might have been a small cluster of neurons, but he still had 8 probes around it to measure the signal from the group of those neurons. So that's one aspect that's important. So 4 brain regions that he ultimately ended up with. There were many sites within each region, so many of these green dots, within the green region. There were 8 electrodes per site, per dot. There were 8 electrodes. And then sites, some of these sites had multiple neurons in them, others had only a single neuron in them. So these are the key ideas around the most important parts of the data that he collected. Now this slide here is a elaboration of exactly this. This is trying to show what the subject saw. So if you think of this one single clock, this is basically what was on their screen. A single clock with a little hand. It's not a second hand, but this hand moves more faster than a second hand. But this little line is rotating around the clock, and there's a dial with numbers around it. And the test subject basically sees this when they start. And what they're told is that this dial will keep moving on its own, independent of what they're doing. But they have to, in their mind, what they have to do is they have to wait until the dial goes around the clock a full 2 times. So it starts off at 12 and it goes around 1 time to 12. It goes a second time to 12. And only after that, the subject is supposed to think about when they want to press the button on the keyboard. Okay, and they can do it, and they were given a very, very large latitude. They could do it immediately after it goes the second time around, which would have been, I think in the experiment, it would have been 2.5 seconds, because this dial went around once about 1 1/4 second, 1.2 something seconds at a time. So after about 2.5 seconds would have been the earliest time that they could have pressed the button, the keyboard button. Or they could have waited as long as they wanted. They could have waited an hour if they wanted to. There was no limit. What they found was that most of the people who ran through the experiment, the maximum they waited were around 19 seconds. That's just the starting, the ending time scale. But there was no limit in the experiment. If somebody wanted to wait 40 minutes, they would have done that. Although I think technically there may have been a limit to how long the data collection could have happened. It probably was on the order of a few minutes. But all of the experimental runs completed within about 19 seconds. That was the maximum that anybody waited. They kept allowing this dial to go round and round and round and round and round and round for 19 seconds, and then they pressed the button. That was the maximum time that anybody waited. So that just gives you an idea of the freedom and the flexibility that the subject had in deciding when to press the button. They really could have done it any way they wanted. The design of the experiment did not dictate what they did or when they did it. So that was one aspect. The second aspect was when they made their decision to press the button, they had to simply keep in mind the number around which this dial was at that time. So what they might have been doing is thinking, well, when it comes around to the 4, I'm going to press the button. So in their mind, they made a decision. Okay, their will was their decision, or not decision. Their pre-planning decision was, when it comes to 4, I'm going to press the button. Okay, and so actually, sorry, let me take that back. What they asked the subjects to do is to decide where the clock was when you made the decision of when you're going to press the button. So there are 2 things here. Okay, one is when you made the decision, when you made your will to press the button, and the second is when you actually press the button. So let's say if the person decides that they're going to press the button when it hits 4, when the dial comes to 4. Okay, but they may make that decision when the dial is on 1. Okay, so there's 2 information here. One is when they will themselves to decide when to press the button, and when they actually press the button. Okay, so their will was around 1, and they actually pressed the button around 4. Those are 2 pieces of information. And when they actually press the button, they don't have to remember as much, because the computer is recording that with a very, very highly accurate clock. All they have to do is they have to remember when they made the decision that I'm going to press the button when it reaches 4. Okay, and they weren't required to do it like, I'm going to press the button when it reaches 4. They didn't even have to think about any particular number. They could have just blindly said, I'm going to press the button, in 3 seconds from now. They didn't have to necessarily say, when a certain number comes, they could have made their decision any number of ways that they wanted. There was no built-in set of rules as to how the subjects behaved in this experiment. The only thing that they had to do, which was subjective, was they had to keep track of where the dial was when they had their volitional idea, their cognitive intention to press the button. When that occurred, is the only thing they had to keep track of, which basically was a visual signal. They had to basically have a visual recollection of where the dial was and keep that visual recollection and maybe encode it in terms of a number, okay? A little bit past 1 or between 1 and 2 or exactly on 6, whatever it is, however they encode that information, they had to simply remember that aspect. And they were asked that. And in in Fried's experiment, these dials are an example of what actually transpired. So the first dial is an example where the dial is rotating and the clock starts. And the second dial is where they actually make the will meaning their decision of when they're going to press the button. So in this case, the person made their decision to press the button at this point between 4 and 5, halfway between 4 and 5. And this is described as a W for will, okay? And the point at which they actually press the button is denoted as P in this case. And that happens to be halfway between 5 and 6, okay? And so the subject was asked that the way they were asked to say when they had their will to press the button was they were allowed to actually move this dial backwards. So in this case, they're moving the dial backwards to where they thought they made their will. Now, as you see here, there can be a difference between when their will exercised and when they move the dial. But the difference in time, and the experiment had many statistical types of calculations which accounted for that. So it was a very sophisticated type of analysis that Fried did, his team did. But this is more accurate because it visually allows a person to physically move. They don't simply have to have a visual record of where the dial happened to be. They actually are moving it, and they can kind of fudge it with it. Well, I think it was here. No, it was a little bit more, and until they find the right spot and they say, okay, this is my answer, and they say that's, and then the system records it, and then during all that, while it was happening, it's recording, neuronal activity. So there are 3 important points in time that was recorded: the 0 time when the clock actually started, the W time when they actually made their decision, when they willed what moment in the future they're going to press the button, and that moment in the future when they actually press the button. Those are 3 time points that were recorded as part of this experiment. Now, there are a lot of things that are in detail in the paper. I'm not going to go into all of that. Again, 4 brain regions, many sites. Actually, this should have been deleted. Sorry, I don't know why that was. So the next slide shows the results of what he found. And this is, if you read the paper, he has a ton of data, he has a ton of analyses, and he supports the ideas that he came across in many, many different ways. It's a very, very strongly done study. He isn't looking simply at the level of the study, he's looking at the rate of rise. He's looking at, so just this one slide I'm going to explain a little bit, but there's many aspects to this, and this slide only goes into the frontal lobe, but he has the data for all the different regions, actually more than the 4 regions. As I said earlier, he collected regions across, I don't remember exactly how many, but it was more than 4 regions across the brain. What he found was that the signal in the frontal lobe was the strongest, okay, as far as the signal of the will. And that's the reason why I have this graph for the frontal lobe. And what he found was that the signal with the frontal lobe was the strongest, but also started the earliest. Now, this is the part that gets into the free will aspect of this discussion. And I've hinted at this in prior talks, but this is where I want to go a little more detail about what the significance of this is. And what this graph basically says is that he found 2 types of neurons. Again, excitatory neurons, which is the red graph, and inhibitory neurons, which is the blue graph, okay? And he found these excitatory and inhibitory neurons all over the place, everywhere. And these excitatory neurons included both single neurons as well as clusters, multiple neurons. And same for the inhibitory. There were some inhibitory neurons, there were single neurons, others there were clusters. But they all acted very, very similarly. There wasn't a whole lot of difference, although there were differences in the signal strength and the rate of firing. Those are more internal aspects, but the data that came out of it, the data showed that the behavior of these neurons were relatively similar. There wasn't a lot different. Now, what you see in this graph is that the excitatory neurons and the inhibitory neurons, they reach their peak, their maximal or minimal point around the same time. Now, on this graph, let me say that this vertical dashed line is the point of W. Okay, this is the point at which the person, and in this graph is a normalized and an aggregated. It's not a simple mean average. It's a complex mathematical averaging of all of the data across all of the subjects, across all of the runs, across all of the different electrodes, all of the different areas of the frontal lobe. It's a huge type of analysis that was done to come up with a single graph. But this single graph shows all of the different complex things that were happening in the brain. And what it shows in summary is that the signal in the brain that was occurring is this, that at the time when the person made their decision, when they decided that they're going to press the button in the future, okay, whenever it is in the future, and this is irrespective of whether they're pressing the button, 0.1 milliseconds in the future or 19 seconds way down the line in the future, that doesn't matter. I mean, they normalized all of that, and they did calculations to change any impact from those variables, so that this graph encapsulates all of the nuances and the differences that they would cause. And then all of the W points were aligned along this vertical line across, again, all the patients, all of the runs. And the finding basically shows, and on the bottom scale is in milliseconds. So this 1,000 milliseconds means second. This 1,000 milliseconds here means a second in the future. So this negative 1,000 milliseconds, this negative 2,000 milliseconds, what you'll see is that this red graph and the blue graph is told him, his team, that there were signals in the brain that began to appear around a little before one and a half seconds before the person before they made a decision to do something. Okay, before their will, and again, by will, we're talking about conscious will. Okay, so before the person had a conscious awareness of making a decision, okay, more than a second and a half before that, parts of their brain started to do things. Okay, and if you go into the data, he goes into a lot more analysis, that he looks at the rate of rise, he looks at the first and second derivative. There are many types of other, he looks at the types of signals, whether they're a pulse train, whether they're sporadic pulses. The actual signals that are recorded are very complex, but he provides all of that. There are many types of graphs. I'm not going to go into it because most people, I don't think, would understand exactly what they mean. I don't want to go into those details, but this graph summarizes the key ideas here, and that is that this red, whether you look at it from the red signal or the blue signal, they're basically saying the same thing. That one point that he didn't do in this experiment, but subsequent people in other experiments did later on, okay, to expand on this work, is that the fact that there was a red signal and a blue signal, meaning an excitatory signal and an inhibitory signal, they took on that and they designed further studies to include additional aspects to a very similar structure, where when they told the person to make a decision, that they were going to press a button, okay, they told them to do that. But then when it came time for them to actually press the button, they gave that person an additional option of either yes, pressing the button, or no, not pressing the button. This is not in Fried's or Libet's experiment. This was in subsequent experiments. But the fact that this excitatory and inhibitory areas of our brain exist, or regions, neurons and clusters of neurons exist, means that a person is able to make a decision but not act on it. And that's, in philosophy, we think of it as the will and the won't, right? And that's a version of this. So this experiment has been further done in more complicated ways. But the gist of this is that more than a second and a half before, and again, this experiment was measuring signals from the neurons itself. In Libet's experiment, because the signals were weaker, he found that the signals that he found were around, the maximum were around 0.4 seconds, a little less than half a second prior to, because again, signal was lost. So he wasn't able to measure the signal strength as much. But in this experiment, he found, Fried found that it was earlier than the second half, that some type of decision started to be made in the person's brain, every person's brain, long before they actually had an awareness of their actual conscious decision being made. And what that indicates, what that suggests, is in subconscious parts of the brain, and the word subconscious I put into quotes, okay, that somehow there were parts of their brain that knew when this person was going to decide, when in the future they're going to press the button, even before this person actually decided when they're going to press the button. So this is a little confusing because there are two things that the person is doing. They're deciding and then they're pressing. Okay, this is only talking about the deciding, because the pressing becomes irrelevant. Whether it's a half a second or 1 second or 19 seconds in the future, it becomes totally irrelevant. The only key point here is when they're deciding when, the W, the will. So W is the will to act. Okay, when they made their decision, when they had their will to act in the future. Okay, again, this found that all the regions had very similar. This graph was rather similar, except the amplitude was different, and the starting point was, this was the best case, the frontal lobe, where it was a little more than a second and a half. In other regions of the brain, it was much abbreviated. From around I think 1.2 seconds down to about 0.2 seconds was in this graph. It was a smaller version of the graph, you can say, in other regions, but this was the most significant and sincere. So what he found was that action, some type of action, occurred in the brain, or some type of decision, will for action occurred in the brain greater than a second and a half prior to when the person's actual conscious awareness of the will occurred, and this occurred in the medial frontal cortex. That's MFC. So even though this was in the frontal lobe, it was in a specific part of the frontal lobe. And the reason why this is important, this medial, is because this task was measuring motor skills, and the medial area of the frontal cortex is closer to the motor cortex. That's why it's important. I won't go further into that, but for the people who understand the neuroscience of it, that's an important aspect. Also, the finding is that what this single graph doesn't really show, but he goes in, elaborates, is that there are multiple preparatory signals all over the brain. This is what's unusual and strange, which was not expected. But when the person's will came into being, that will was all over their brain. Okay, it wasn't just in one place. That's what he found. So that's what this last bullet here says, is that the brain had multiple preparatory, this rising and the falling, these two blue and red signals. There were multiple copies of this, scattered throughout the brain, slightly shifted forward or backward, in time, and amplitude higher or lower. But this same graph was replicated throughout the brain in many different areas. And lastly, what this suggests is that there's, again, subconscious in quotes, there's some type of subconscious action that began to occur in the brain long before the person had any kind of awareness, any kind of, actual conscious, understanding that they are making a decision, they're going to make a decision. That's the strange part of it. But subsequent to this and subsequent to other experiments that followed on, all of the data and the research from all of these experiments together are what leads us today, and many neuroscientists today who have followed the history of all of these studies and looked into detail and challenged, etc., they tend to feel that this concept of free will really is not there. We don't have free will. Most of the strongest people who the higher level scientists who not only analyzed these outcomes, but they've done further other types of studies, they're very, very staunchly in the camp that free will is a illusion. And actually, there are other types of experiments which have tried to figure out why it is that we have this concept of free will. And I didn't go into this here. I thought about doing it, but it would have gone into a different direction. Maybe I'll do that later. But there are actually studies that have gone into trying to understand why we have this feeling, this emotion of free will. And there's actually several studies. There's a neuroscientist, Weber, who did some very pivotal studies. He was actually a psychologist, but he went into the computational side of psychology, a little bit into the neuroscience, and he did some very, very interesting work where he basically came up with, a lot of his research concluded that this concept of free will is an imagined concept that we all have. And the reason behind it, there are multiple reasons that he hypothesized, and there's still research now going on, building further from the ideas that he laid forward. But this concept of free will is an imagined idea that our brain concocts for many reasons. But basically, it gives us a reason because we need to have a feeling of agency. In psychology, those of you who understand what agency is, when we feel like something happens randomly to us and we don't have a say in it, it disturbs us. It gives us an uneasiness. It gives us a bad feeling inside. And he believes, and some of the research does show this, supports it, that this feeling of free will predating when it actually starts, is a figment of our brain. Our brain intentionally does it. It fools itself in a sense, or our conscious mind fools itself. It believes that we have this strong, controllable will that we control completely when we want to do things. In reality, it's not that we control it. In reality, lower parts of our mind, subconscious mind, does things on its own. And once it does things, then our conscious part of our mind basically comes up with an explanation after the fact. I'm going to give one example of that shows when this happens is that and this has been done in many different ways, but one surgery that was done, that surgery itself has been repeated a few times, but it's a difficult surgery. But what was done was that the patient was under, the body was under anesthesia, but anesthesia doesn't cause the brain to shut down. It causes the pain sensation and nerve signals to shut down in the rest of the body, but the brain was still active. So while the person was under anesthesia, the neurosurgeon, they had their scalp open, and the neurosurgeon basically went with a probe and sent a signal, activated certain areas of their brain. Okay, and what they were doing were they were causing certain muscles in that person's body to twitch. Okay, and what they found was that when they caused specific muscles, and this happened, it didn't matter what muscle it was. Every time they did this, when they caused the muscle to twitch, you have to remember that the neurosurgeon was causing the muscle to twitch. That person, the patient who was on the operating table, was not causing any of it. And they didn't even know that their muscle would be twitching. They didn't know which muscle would be twitching. So they were basically surprised. Okay. And if you think about it, think about this way: if you're lying on an operating table and your body twitches, right, most of us would be embarrassed by that. Okay. And that's a key aspect to understand. I'm going to explain what happened next. When the neurosurgeon activated, and this is irrespective of what part of their body, what muscle they activated, when they activated a part of their body that caused the muscle to twitch, and then they asked asked that subject, the patient, did you do that? The patient always answered yes, okay? But then they asked the patient, why did you do that? And the patient always gave a made up answer, okay? And the experiment was a little more complicated so that the neurosurgeon could kind of tell when the patient was making up versus not making up these reasons. And also, when the neurosurgeon caused something that was embarrassing to happen, it was more telling because the person's emotions and other things came out. So basically, what came out of that one experiment, again, which was repeated, was that the person had no awareness or control over their body. Their body was being made to do something which made them uncomfortable. And their conscious mind always rationalized that uncomfortable feeling. And they communicated to the neurosurgeon that, yes, I'm under control, I'm doing all of this, and I'm doing it for whatever reason. Even though if the reasons were silly, they still gave a silly reason, because to them, giving a silly reason still was more comforting than this feeling that you're out of control over your body. Your body is twitching. So that's one example,
Sanjay,
of what we believe.
Sanjay, I want to leave some time for Q&A. It's already.
This is the end of it. That was the last slide. Yeah.
Wonderful, wonderful. All right, folks. So it's time for questions. Let's collect all the questions, and then we will go ahead and have answers. So go ahead and type exclamation mark or raise your hand in Zoom in order to ask questions. Give me just a second here. We'll start with Mr. Kranz. Sean, go ahead.
Hi. First, a quick comment. I always keep those two studies here right within arm's length. And they're not too thick if anyone actually wants to go through and read them for themselves. They're a little technical, but they're actually, as you can see, now this is Libé in 1985, and this is Fried in 2011. So they're readily available on the internet, because that's where I got them, and I just printed them off, and they're really interesting. Yeah, the critical point, and you actually, Sanjay, you got to it in the very end there. It was a really good lecture, is the fact that they could easily see by the electrodes and the button press with the finger how long that took. But the interesting thing is the fact that the decision to press the finger was in the middle of that stream from the electrodes, showing that there were neural activity increasing to the with regard to the finger. So the increasing happened and the finger went and pressed. The interesting thing is the neural activity was actually before the person decided, oh, I'm going to press the button now. So your brain is like, or your will is like halfway between the neurons getting excited and your finger reaching out and pressing the button.
Wonderful.
I just thought I'd simplify it by just showing my head in between the two.
Excellent.
Thank you.
Thank you. Thank you. Next up is going to be Claudio, Joe, Linda, and Vanessa. Claudio.
Yes, thank you. My question is, you mentioned the unconscious mind playing a huge role in the free will with the inhibitory neurons and the excitatory neuron. What about genes, genetic information like DNA? Can they also play a huge role? I believe they do, right? Can they influence those two types of neurons?
Wonderful. Excellent. So what is the role of genetics in so are you saying I just want to make sure I understand what you're saying. Are you saying like genetic predisposition or genes on the fly?
Yeah, genetic dispositions.
Okay,
for example,
very good.
genes inherited.
Wonderful. Wonderful. Excellent. So is there a genetic predisposition? Next up is going to be Joe, Linda, and Vanessa. Anybody else has questions, go ahead and type exclamation mark. Joe, go ahead.
Yeah, so I kind of have like a basic question. Like, what would someone have to do to prove free will exists? What kind of experiment would you have to conduct? Like, what would be the evidence that needs to be, shown?
Wonderful. What would experiment to prove free will look like? Next up is Linda.
Sandra, I was wondering if you'd want to make any comments about the impact of chemical imbalances in the body, or even nutrition.
Wonderful. Chemical imbalances and nutrition. Okay, I'm going to put it with genetics in terms of various factors. Excellent. Next up is going to be Jim. Jim, go ahead. Vanessa first. Vanessa, what's your question?
Okay, I'm inventing a term. Is it possible there's something like restrained free will, where maybe your desires may suppress some synapses from either firing in the neurons? Like, for example, you're getting dizzy, you're a dancer, and like the memory triggers, a focal point will get my balance so I don't get dizzy. So somehow your desires may limit some neurons from firing, so you have some influence over what decision might ultimately be made or signals sent out. Is that possible?
Wonderful. Do desires.
Did I understand? Did I explain that well enough? Or
yeah, I'm going to put it as do desires limit free will in some way. Next up is going to be Jim. Jim, go ahead.
Yeah, I would ask the question, is free will just a linguistic problem? I happen to think it is, because we've couched it in a misleading way.
Sure.
We've couched it as determinism is the opposite of free will. That's not the case.
Sure. Let me go. Let's put it as a question for everybody, and then let's discuss it.
Yeah. Okay.
Is it a linguistic problem?
Is it a semantic problem?
Semantic problem. Okay, very good.
Thank you.
All right. Next up is going to be Madeline, followed by Cam. Madeline, what's your question?
Thank you, Sanjay. This is fantastic. And I liked what you said about free will being an imaginative construct related to agency. And it seems to me that these experiments haven't removed free will. They've just sort of taken it from the tiny tip of the iceberg that was showing to a much larger complex of icebergs underneath the surface, so to speak. And I'm wondering what the thinking is on those interacting networks still being some form of free will.
Very good. Excellent question. Interacting networks. Do these experiments simply move the problem to it at another place? Next up is going to be Cam, followed by Mike. Cam, what's your question?
Yes, it seems to me that the whole discussion presupposes, or rather fixes definition of free will and determinism to be opposite of each other. And as such, then the whole discussion proceeds to say that if determinism is true, at least as we are concerned in regard to the scale of human behaviors, if determinism is true, therefore free will would be not true. But it's by assuming that those words are opposite of each other from the beginning. So it's like it's a presupposition.
Sure.
And is it really how we use free will in day-to-day life? It seems that from what I know, there are some studies that show that most people don't mean that when they talk about free will, they are not talking about uncaused behavior. They talk about something else. And therefore, this something else might be or not be compatible with behavioral determinism. But
Thank you. Thank you. Cam, I'm going to combine that with James' question about linguistic. How are we defining these terms?
Yes,
So we'll put it together with that. Thank you. Next up is going to be Mike. Mike, what's your question?
My question is, does Mr. Kranz's collection of papers make some of this moot? Because LeBay's paper was from in 1939, and that started all this stuff off. One of his first papers, and now Sugar in 2018 came up with something that said, and I guess Kranz is holding up Sugar's paper now, I think. It said that it's all part of the same mind. Just because we're not aware of it, which is what Madeline kind of said, it's all the same mind. Just because we're not aware of it, and just because the prefrontal cortex can cancel that decision and create some other kinds of noise. But we should say that it's a very interesting result. And Lebay won what we call the virtual Nobel Prize in psychology. And I don't know if that was
you want to put a question down?
The question is, does Kranz's discovery of papers make some of this moot? However, it's still an interesting point. Then I would say one more question.
No, let's as long as it's a question.
One more question. The fact that quantum mechanics changes the entire concept of what causality is, that the effect can come out before the cause and still be all right.
Wonderful.
Does that have a bearing on this discussion?
Thank you. Thank you, Mike. Let's look at all the questions. I've got several questions, and let's see what's the best thing to do. How about Sanjay? You give short answers to the question, and then people can chime in, or would you like other people to answer? I would like you to answer in short, and then if anybody wants any follow-ups, folks, keep track of things you want to talk about. We'll just run through all these questions very fast, get Sanjay's answers, and then open it up for comments for everybody. So first one is in terms of the role that either genetics or chemical imbalance play in all of this. Any thoughts on that?
Yeah. So, obviously, everything that happens in our brain is affected by, there's two ways to look at this: nature and nurture. But the modern concept is actually not it's nature or nurture. It's actually not that it's both. It is both, but it's not that we have extremes. It's that it's a continuum. And there's actually, this is the most complicated part, is that through epigenetics, what we've learned is that whatever has happened in whatever happens through predisposition, meaning nature, the purely genetic aspect of our brain and its formation, the topology of the network, and the types of neurons that we have in different regions, and the length of the axon, all the interconnections, everything about the brain, it derives directly from DNA. That has a lot of effect. Also, the nurture side of it, where depending on the experiences we have in life, the nutrition that we have, I think Linda asked about nutritional. If the person has a disease, if they have chemical imbalances, all these things, would arise from either nurture or nature. They can come from either of these. You can have toxins that come from the environment, which cause chemical imbalances or that cause damage in the brain. So it's very complicated, but basically, two parts that we have to look at is that during growth in childhood years, the brain is in this chemical soup of the world that the child lives in, right? They're eating, they're drinking, they're breathing in all these chemicals that come into their body. Most of those chemicals are good. They have to be nutritional or oxygenation, etc. But there are, in some cases, bad chemicals which cause damage in their mind. So both nature and nurture lead to structure and formation and the functional behavior of the brain in certain ways. And that is driven by both nature and nurture. Then there's a whole different layer called epigenetics, which after they're born and living and into throughout our adulthood, epigenetics is another aspect of our DNA, where what it does is and this doesn't apply only to neurons and neuronal cells, it applies to every type of cell in our body. It changes the way our cells behave based on the real world environment situation we're in, not just real world in terms of the physical world, but our psychological world, our physiological makeup in our body. So if our blood pressure is high, that high blood pressure would cause epigenetic changes driven by our DNA, again, which would be based on partly predisposition, partly based on our biological condition at the time, partly based on our nutrition. All these things put together would cause different both functional and structural changes in the brain. So it's a very complicated, but all those things play together.
Wonderful. Next question was the question from Vanessa. What roles do desires or values play in kind of limiting free will, or what role do they play?
So I was trying to think about examples that I can give that were realistic examples of, free will that most sophisticated people, enlist in our everyday lives. The problem with using those types of examples is that they are so complicated and they deal with so many internal aspects of our brain that, I would have to explain, or we would all have to understand the interaction between all those different aspects. I mean, just earlier in the talk, I said things which I later realized that I understood what I was saying, but some people might not have understood. For example, when I was talking about the information, the signals in the brain, I understood that the signals also come from within the brain, but other people didn't. So similarly, there are so many other aspects to free will and the matrix of events and conditions and states that exist within different subregions of the brain. So when we're talking about desires and values for free will, desires and values are a form of information, okay, and they are encoded in signals. For example, desire, we often think of as emotional, right? That you want something, you desire something. It's usually a positive, but a desire can, in terms of neurology, in terms of neuroscience, it can be negative also. That's something that is aversive to you. It's an aversion, right? It can be a positive or a negative, but we would call it the same way. And values would be states of mind, you might say, that you value certain situations and certain environments, certain types of behaviors in other people, certain conditions that you happen to exist in. For example, you might value rain, okay? Other people would not value rain, right? So it's complicated, but basically what it comes down to is, how these desires and values, how they're encoded in a specific person's mind, the strength of the signals that they engender, and the amount of distribution of those signals, of that information from the various parts of the brain, how much they proliferate throughout the brain and where they end up, and how they combine, right? All of that together, it's extremely complicated, will ultimately go into, there'll be inputs into any free will, any volitional decision that you would be making.
Wonderful. So folks, what I'm going to do is that as soon as we're done, we're going to keep the answers short. As soon as we're done with the answers, I want to give a chance for everybody to talk about how they think about this issue. You'll get 1 minute. Okay. I'm going to start stopwatch, and you can talk for 1 minute about where you stand on it, how you think about it. Okay. But let's finish the questions. And the next question was Madeline's question. It's very interesting. You traced free will, kind of the experience of free will into, this movement over time and across the brain. So it's kind of moved from like a decision to this entire network. So is it just moving the issue to a different part, more complex part? How do you think of that?
Okay, so I don't think it's simply moving into another part, because the key idea that we have to remember in free will is the idea of free, right? The idea of choice. And when we're talking about choice or free, your decision, your ability to do or not do, that gets into consciousness, that you have to be conscious to do it. However, a lot of these types of things that we're talking about, and a lot of the things, actually, if we break down and analyze it even further detail, we will find that things that appear to be like free will happen all the time in our subconscious, but they get masked or they come out in our behavior in other ways that we don't characterize to be free will, but they actually are. Okay, there are many things that happen all the time like that anyway, but we only pick and choose those things that we want to have control over, and we call those free will. So this is not simply where we're moving it further down the scale, further deeper down, because things that are further deeper down are outside of our conscious awareness. That's the key idea here. Again, the concept of free in free will is the key word, that once it becomes opaque to us, once it becomes hidden from us, from our awareness, then it becomes automatic, right? And that's what this discussion and these ideas around this research is really how much of our behavior is automatic versus how much of it is chosen by us as a being. So I don't think it is that it simply moves it further down. It moves it down into the realm of no longer being free, of being automatic. Okay. And that's what most neuroscientists believe.
Wonderful. Next up is the questions that Jim and Cam asked. Is this merely a linguistic issue? The way in which we define terms like free will or determinism?
I mean, one way to say it is yes. I mean, it is, but there's, well, first, the problem is if we're talking about, I mean, we're going a lot more into the philosophical side of things. Now, the challenge when we're talking about anything, looking at it from the broad views of philosophy, is that what one person has in their mind about a notion and what another person has in their mind about the same notion, even if they're using the same words to describe them, they may actually be looking at them in very different ways. So the words that we use don't elucidate what's actually the concepts and the complex understanding that we have about a topic. Okay? The words don't convey much. And the words that we have to convey are sometimes limited. For example, this is why when you look into the philosophy of determinism versus free will, there are compatibilism and incompatibilism, and there's so many different subcategories along this continuum that we call determinism versus free will. There's so many subcategories, and it's not just subcategories. It's actually that this continuum, this line, actually becomes orthogonal to itself. There's so many other orthogonal lines to it that it becomes very difficult to talk about. And this is the reason why in philosophy we don't have answers. And most people, they basically have their opinion, they have their ideas, and they stick hard and fast to it. It's very difficult to budge because you can't prove that the idea that you have in your mind is the same thing that another person, even if you agree to have the same ideas, in fact, what's inside your mind may not be the same idea, but it may appear to you because you're using similar words and you're using similar examples, but there may be an example that neither of you have thought of, that maybe in a year, one of you thinks of this example, which shows there's a stark difference between your views, but you haven't realized it yet. So in one sense, you can say it is linguistic, but I think that it's much more because our concept of what happens inside our brain is conceptual, and we have no way of communicating the extremely detailed concepts. Our languages aren't rich enough.
Wonderful. We'll take one last question, and then folks, go ahead and type exclamation mark or raise your hand in Zoom if you'd like to make 60-second statement of what you think about this topic. So Sanjay, the last question is from Joe. What would a proof of free will look like?
Yeah, very interesting question. I don't know. I mean, that's not something I can give an easy answer to. I mean, I can conjecture, but the challenge is that, well, I mean, so if you want to prove free will, right? What you're saying is that you want to prove that. I'm trying to find the right words, because in the previous question, this is exactly the problem. The words that we have to talk about these concepts, the words are limited, and any word that I use probably will introduce other notions in your mind, which I didn't mean to. So this is a challenge. I'm trying to be careful about how I say it. So whatever experiment we can come up with to prove free will, I mean, it's going to have to show that. I mean, I don't. I mean, this is really difficult to.
Let's table this. That's fine.
Give me a few minutes. I'll
sure. Sounds good. Yeah. Sounds good. All right, folks. So now, what do you guys think about this question of free will and determinism? Anything that has been said? I'm going to start a clock of 10 seconds and not 10, sorry, 60 seconds. I'll give you a 10-second warning in the chat, and then 60 seconds is a hard limit, so we can get as many people in as possible. So first one is going to be Avo, Madeline, and Mr. Kranz. So Avo, go ahead.
Yeah, it's actually a question related to the last question. I have a question related, maybe a simpler version of the topic just addressed. And that is, if we conduct any experiment, whether it's 2 or 3 neurons or 2 or 3,000 neurons, if we repeat the experiment with exactly the same input, is the output always the same?
Thank you. Thank you. But we won't take questions right now. We'll just take comments. Next up is going to be Madeline, followed by Kranz. Madeline.
Okay, thank you. This seems to be a really lovely coincidence. I had just been reading the chapter on Aristotle's physics in Bertrand Russell's History of Western Philosophy. He says, to understand the views of Aristotle, as of most Greeks on physics, it is necessary to apprehend their imaginative background. So this is what relates to what we've been talking about. Every philosopher, in addition to the formal system which he offers to the world, has another much simpler of which he may be quite unaware. If he is aware of it, he probably realizes that it won't quite do. He therefore conceals it and sets forth something more sophisticated, which he believes because it is like his crude system, but which he asks others to accept because he thinks he has made it such as cannot be disproved. The sophistication comes in by way of refutation of refutations, But this alone will never give a positive result. It shows at best that a theory may be true, not that it must be. The positive result, however little the philosopher may realize it, is due to his imaginative preconceptions, or to what Santayana calls animal faith.
Thank you.
So those were the imaginative preconceptions.
Wonderful. Thank you. Thank you, Madeline. Appreciate that. Next up is Mr. Krantz.
I have here the medical physiology, Guyton's medical physiology, which doctors all study to get a clear idea of how humans work. And on the first page, Guyton writes, the human being is actually an automaton. And the fact that we are sensing, feeling, and knowledgeable beings is part of this automatic sequence of life. So I just thought I'd throw that in. I'm a hardcore determinist, like hardcore determinist, not soft determinism. I think our thoughts, the very conversation we're having right now, are completely determined by physics. Thank you.
Thank you. Next up is going to be Joe, followed by Vanessa. Joe.
I'm a bit of a soft determinist there. So, that's my perspective on it, is that, I kind of like the way Viktor Frankl approaches this topic, in the sense that between the stimulus and the response, that there's a space that we can actually say no, and so that we do have the will to actually stop and make a decision. And I think those are called the inhibitory neurons, as you're referring to, the red neurons that we were talking about a little bit earlier. So that's where I stand, is that we do have some control over our decisions, and we do have to behave as if we do have free will.
Wonderful. Thank you, Joe. Next up is Vanessa.
Thanks, Sanjay.
Yeah, I think it might be a hybrid of the two. I kind of think of the brain maybe like a little toddler. It wants it a certain way, but more for self-preservation. Likewise, if you're getting stressed out or something, it may suppress some of the neurons. So whether it's just realizing, okay, my blood pressure's going up, I got to calm down. So maybe, that brain may be realizing, okay, I got to kind of protect myself here. And just like there are repercussions, like realize we don't live in the world by ourselves. So my brain may want to do something, and I realize, wait a minute, if I were to go and do that, maybe we need that override just for not to get so furious and smack your neighbor around or something. I think there's a combination of the two. It could be they just have discovered it, like you mentioned, all that information that's being put in there that the brain is processing. I mean, it's kind of hard to map that out. And by the time we figure it out, there'll be a new problem.
Wonderful.
But
Thank you.
Very good talk. So, continue. Really enjoyed it. Thank you, Sanjay, and everybody.
Thank you, Vanessa. Next up is Maritza.
So I think it's a fascinating topic and question, the idea of, determinism, looking at it from a view of the neurons. It makes me think, I don't think about breathing every day, but if I should choose to, I could force myself to breathe deeper, to breathe harder. So I feel like somehow, somewhere within me, there's a little bit of both going on, because breathing happens whether or not I think about it. So that's happening for me. I'm not consciously thinking about it, but I can exert energy in my consciousness to change my breathing pattern if I wish to. So I think that it's that question of, it's not a question of either or. There has to be some middle ground, and I'm intrigued by, as we get more and more technology, as we're seeing more, that there's science to point the hows and the whys for us. Thank you, Sanjay.
Wonderful. Thank you. Thank you, Maritza. Next up is Claudio, followed by Jim. Claudio.
Yes, I tend to be a compatibilist. I tend to be I know there's studies that have proven that our behavior, future behavior, are influenced by our genetic, let's say 45% and 55% by your environment, especially the environment that we encountered during our childhood. they influence us later, right? But you know, not everyone becomes an alcoholic if both their parents are alcoholics, right? There's risk factors, but risk factors do not necessarily mean they're actually gonna become facts, right? Facts of life. They're potentials, but not actualities. Not everyone who has a brain tumor will become a compulsive gambler or, let's say, a mass shooter, right? There's things that can overcome your predispositions, like mindfulness, meditation, and other things that society provides. Yeah, so I tend to be a compatibilist. I don't actually, not a hard determinist or anything like that. Thank you.
Thank you. Okay, I forgot Mike. So it's going to be Mike followed by Jim. Mike.
Well, I don't have an opinion on it. However, Lebet said that his thesis was not a conclusive answer to the problem of free will. Lebet, not me. Lebet believed the brain was a deterministic device, and he said he quoted Robert Frost in this. We do the steps that we've been shown by everyone we've ever known until the dance becomes our own. And so we can say that we haven't solved the problem of free will, but it isn't a question of the bigger problem is whether the brain is deterministic rather than we're following a script, as Shakespeare said, that life is but a stage and we're just actors on the stage, and some evil demon like Descartes is running this show for us.
Thank you. Next up is Jim.
The problem of free will is a linguistic confusion. The opposite of determinism is not free will. The opposite of determinism is random chaos. The opposite of free will is not determinism. The opposite of free will is coercion. When we say a person is acting freely, we mean that they are not coerced. They are not slaves. They are not prisoners. They are not being robbed by a person with a gun. The opposite of free will is coercion. We often have times when we are coerced. Children are coerced by their parents. Children don't get free will. Parents coerce them. It's as simple as that. All of our behavior is determined. We don't control our heredity. We don't control our environment. We don't control the language we speak or the religion we grew up into. We are deterministic beings who act freely because we are not coerced. That doesn't mean our behavior is uncaused. Our behavior is always caused, usually by things that we have no control over. You can't decide whether something is going to taste good to you or taste bad to you. It's an automatic response of your taste buds and what you eat and your history.
Wonderful.
So it's just a linguistic confusion, and we shouldn't be so concerned about it. There are times when we are free. Whenever we do what we want to do, we are free, but we are always having our behavior being caused by forces outside of ourselves.
Thank you.
That's it.
Thank you. All right, folks. This was a great meetup. Sanjay, do you know what you're going to do next?
No, I usually don't think. I mean, it depends on which areas I think are important for folks. Like I think about a few steps, a few talks ahead, and then what do I need to do intervene?
Sounds good. Sounds good. So, folks, I want to tell you about some things that are coming up next week. 1 week from today. Go ahead, Sanjay.
Just one. The question that Joe had asked earlier about experiment to.
Oh yes, yeah. Go ahead.
I mean, I thought about it, and I think it's possible, but it's not easy to do. Basically, to show that free will does exist, you basically would have to show that information does not follow a deterministic path, which means it does not follow a network. And so in terms of the brain, you'd have to show that information in our brain flows not simply through neurons, it flows through other methods, for example, through the interstitial space, through the neurotransmitters that we have. I mean, neurotransmitters do carry information, but you'd have to show a systemic model of neurotransmitters augmenting and perhaps supplanting the electrical signal from neurons, and maybe something else. I mean, I don't think quantum mechanics answers it, but you'd have to say, you have to find some other way the information is propagates through.
Thank you, Sanjay. So let me tell you, folks, about what is coming up. So next Wednesday, I'm going to do a meetup on the interaction between writing and conversation, because that is actually at the heart of what we are doing here at 52 Living Ideas. Typically, we take works, written works, and we talk about it in great amount of depth. And what does that do? The new idea that I'm working with Mark Stallman is the idea of what is called a scribal hyperbook. So if you look at the scribal age, whether in India or in the West, whether you can look at the peripatetic school of Aristotle, you can look at Aquinas' lectures that we're studying in University of Paris. The way it worked is that you were studying some book and you were surrounded by people. Books were hard to come by. Written work was hard to come by. So there would be all these people focused on the same written work, talking about it. And this envelope, this is a point that Walter Ong makes, this relationship between orality and literacy. He looks at the envelope that conversation puts around a written work. And that's what we are doing here in the meetups. But a crucial part of it is to take it and put it back into writing. So all the effort, all the work that is being done orally, go ahead and put it back into writing, and then continue this loop between written and oral. So we're doing the experiment with Mark Stallman on his series on digital Catholic social teaching. That's on Sundays at 3 p.m. I'm also starting to do that with Aquinas that we are studying. But the most ambitious thing I would like to do is to do this with our Thursday meetups. We are launching a Thursday meetup. These are all on 52 Living Ideas. So if you're coming here from the Greater Philadelphia Thinking Society, many of the meetups I'm talking about are there only in 52 Living Ideas. So tomorrow we are going to do a meetup on crowdsourcing self-improvement, where everybody talks about one idea that they have found has transformed their life. Okay, you get between 2 to 5 minutes to talk about any one idea. There is a featured presentation by John, which is going to be incredible. He's calling it the measure of man. How do you keep growing? How do you measure your rate of growth? How do you measure when you're stagnating? What kind of standards you set for yourself in order to keep growing? That's what he's going to be talking about. But that'll be only about 10 to 15-minute presentation, and then everybody else gets to talk about their ideas. I want to figure out a way of putting that back into writing so people can talk about it, interact with it in a written format when they are not in the middle of the meetup, and keep building that. So I want to look at this interaction, and what I will do next Wednesday is to talk about it at a more abstract level. We will have the experience of Thursday, Sunday, and Monday of actually doing it. I'm going to come up with like blog posts for each of them. I have blog posts for each of them, where we will be putting material that you can read beforehand and come. And whenever we do any meetup, we'll put the video back there. So again, the loop between meetups and videos, meetups and writing, I want to keep building that up, and also make it easy for people to interact through comments and things like that. So please join us in these experiments. And then the week after, so as far as Comprehensivist Wednesdays go, next week is going to be interaction, this virtuous cycle between writing and conversation. The week after that, Mike is going to be back. The week after that, CJ will be back, and then it'll be Sanjay coming back. All right, folks. So if anybody has any comments about the hyperbook or anything about interaction between writing and especially people who are regulars here, like Joe, Marisa, and other people, any thoughts about what do you think of this idea of taking what we do and putting into writing? For example, the Aquinas that we are studying, actually what we have are class notes of a guy called Reginald, who was his secretary, who would keep taking notes, and that's what we have. That's all we have, and it's actually fantastic. So that way of taking oral presentation and putting it into words, because then people and we are talking about it now, so it's like going back and forth between those things. One of the insights I have is that structuring this around questions is a very powerful way of doing that, because what it does is that it not only presents, it breaks what somebody's presenting into saying, okay, these are the questions I'm trying to answer, but others can come in and say, no, I think of it this way. I would answer the question this way. So it makes it easy to break the conversation up into parts around questions. So that is the structure that I'm using for the book. Just to give you an example, we are doing the Gospel of John. We are doing just John 1:1. Okay, John 1:1. So John 1:1 goes, in the beginning was the Word, and the Word was with God, and the Word was God. He was in the beginning with God. That's it. Aquinas has a 30-page lecture on it. I have shortened it to about 6 pages, I think 7 pages, something like that. So we have, so what we'll be doing is that we'll be coming up with questions about what existed in the beginning. What does the term word mean? What does the term principium, which is translated as in the beginning, but principium is a far more powerful word, having all kinds of meanings in Greece, like the source or principle, all of that come from it. What is the meaning of that? What does it mean to say to be with something as opposed to in something? Why is it that John begins the way, what is it about John, the Gospel of John, that it begins the way it does, which is very, very different from other Gospels? And why is it that he's focused on the Word rather than, for example, God or other things? So why is that? So those are the questions around which I'm going to take, I've taken Aquinas' lecture and I've cast it around those questions. So that's what I'm going to do for this, but I want to be able to do that for everything, and see what this loop looks like. I think we will end up, I'm hoping that it will create a new medium that is a little bit more powerful than the Zoom meetups themselves, in combination with, currently we have the writing in the meetup and we have the Zoom, which is live. But I think what this will do is that it will scale up the writing, which will capture some of the grandeur of what we end up doing live and make it more accessible to more people and create kind of more loops on that. So any comments on that, Joe, Maritza, or anybody else? Sanjay, what do you think?
It's wonderful. It's a very large. It's going to take time, but it's a definitely you're integrating. Trying to think of how to say that. You're integrating obviously the oral, the intellectual, the written forms, but I'm not sure if it sounds like you also want to do this live or aspects of it live. So if that's the case, if you are doing it also in person, then it's going to be a multi-sensory experience.
Yes. Yes. Yes.
which is the best way to learn.
Yes, absolutely. And that's what it was like in Scribble. Like if you look at the Indian tradition, you would be in the home of the teacher, interacting with the teacher and other people. And you would have that entire context around as you try to master something. So I'm trying to recreate that. I'm trying to recreate that way of deeply engaging with written material.
Yeah, when I was young, I actually experienced, probably not the same way that it was, 100 or even 1,000 years ago, but in India, there are places where you have teachers who teach in a multidisciplinary way, and it's fascinating. It's nothing like school that we're used to in the West.
Exactly. Joe, what do you think?
No, I think it's really ambitious, and I think it's got a huge amount of potential. Specifically, I think it will make people more precise in their thinking and their words that they choose. And I really like the aspect of asking questions, the way that questions are going to be structured in this. I think that that's going to allow people to really kind of focus in on where the gaps are with what their learning is, but also, to become a little bit more precise with their words. And I think that adds a lot of value, because once you can reduce, something to what you just have to say, that's very powerful. If you can say something simply and convey something that's very complex. And I think that medium actually provides an opportunity for people to enhance those skills.
Wonderful. Thank you. Maritza, do you have any comments?
No, I really like what Joe just said. I agree with that. The idea of, saying less with more. I think it's a skill that we are called to practice and exercise strongly in this group specifically because, to be generous with everyone's time, we want to say what we are looking to say as succinctly as possible. And I really like that Joe pointed that out. That's a great thing. For the idea of the hyperbook, are you thinking of it literally being a book? I think I was not clear on that. Is it going to be a separate medium?
It's going to be an internet. I mean, so it's several things. So the beautiful thing about the internet is the hyperlink. We take it for granted, but it's such a powerful thing that you click on it, and on one hand, it'll take you to the meetup that you can attend. On one hand, you can take it to the YouTube video. On the other hand, you can go to the original version of Aquinas commentary. On the other hand, you can go to the shortened version of commentary that I have prepared. On the other hand, you can go to a list of questions that will make you think about it. Then I will take the questions, and then I'll take YouTube videos that we do and break it up into questions. So you can just click on saying, okay, I want to answer to this question. It goes directly to that question.
That's super cool.
So it's like, it's a way of kind of indexing all the videos through the text. And then there will be comments, so people can keep adding. The thing that I'm really hoping for, I mean, for example, for Mark, what I have is I have Peter Berkman, who's amazingly good and who knows the field very well. So he's going to play Reginald. So Mark is going to just give the lecture, and Peter is going to keep notes, are going to make notes. And we will be publishing the notes along with Peter's, along with Mark's video. Now that's very powerful because you can go between, because these are not like YouTube transcripts, which are mechanical. This is some human consciousness taking in what is being said and trying to put it down as comprehensively, as organized a way as possible. And what that is going to do is that is going to create more interaction. And so it will be, so what the page would look like is that there will be the video, there will be the entire transcript, or, not transcript, but lecture notes by Peter. There will be separate, I have also asked Mark to divide everything by questions. So he's using slides. Each slide has just one question and then bullet points. Okay, that's what all the slides are. I'm going to divide the videos along that. So there'll be like questions. So, and we'll put slides, the slide images, as a way of indexing that place in the video. So you would have like a bunch of slides on the page where you can say, oh, these are all the bullet points. So you're getting things at the bullet point level. Then you just click on it, you see the video. You click on it, you see the lecture notes. So you have this and then you can interact with all of these so that, and I'll keep track of what people are saying in the comments. And maybe I will get Mark to come back and answer all those questions that are coming up. So we'll keep adding content to it. The same thing I'm hoping to do with the, with Aquinas, same thing with the self-improvement, which is going to be much harder because that's like truly crowdsourced. So that's the most challenging thing. And I want to do this all in parallel so that we will learn from one project to the other. Each of them has its strengths. Each of them has its weaknesses. And what I'm looking for is I'm looking for scribes. I'm looking for people who can keep notes of any kind, in whichever way. You can put it as a summary or you can put it as detailed notes and just send me the notes and I can put it all together on the page, or you can put it directly as comments yourself, or I can give you access to the blog or anything so that we can have this kind of back and forth, the written thing going too. Thanks. Joe, go ahead.
So really quickly, how would you approach and anybody that hasn't seen it, there's an individual that Nick, with your interviews with Nick.
Yeah.
How would that actually work in with this medium?
That what I'm doing, I said, see what I found, and this is something that I tried to do. That's an excellent question. It is much better to do it beforehand. That's what I'm doing with Mark's course. That's what I'm doing with Aquinas right now. Like, because after the meetup is done, after the thing is done, is somehow to cast it back into this medium is tough. So I'm always saying that I want to do it up front. So for example, for the meetup coming up tomorrow, the structure is already set. It's going to be a bunch of ideas. I don't know which ideas, but a bunch of ideas. For example, for Aquinas, the structure is already set. So I have those 6 questions. I've broken the entire presentation of Aquinas along those just 6 questions. Mark has maybe 8 slides. So it's already there. Now, with something like what Nick has done, I'm just putting that as a resource on the page. So if you go to the page, I will, Joe had sent
I can
it.
do it.
Can you go ahead and put that? So you guys, you can see this is something I've put together very quickly. My first attempt at it in 20 minutes before this meetup. So it is very rough, but it gives you a very good idea of what
wait a minute.
Both of them. You can put both of them. The first one is like the overview of the Aquinas John 1. And the second one is the specific meetup we are doing on Monday on John 1:1. So that is just that, in the beginning part. And so if you go to the website, you will find that you can, I put everything there. So you can find like, that's the lecture one. So that's, I have given an introductory video about it. I have excerpted I have my excerpts of Aquinas lectures, the ones that I'm actually going to use in the presentation. So that's all there. In the next one that Joe is putting up, that gives an overview of John 1. So I have 2 interviews by Nick, which are just fantastic. He's very, very deep student of John. He actually learned to read that version of Greek. So he reads it in original, and he has very deep comments on that. I also have Baron reading out the whole thing, so you can listen to it if you want. I have Aquina's link to Aquina's original commentary, the entire lectures. So you can find all of that right there. So that's it. So it'll be like resources, so far more comprehensive resources. I'll be able to put in all these slides. So for example, the great presentation that Sanjay did today, we can provide it, afterwards or before, or we can put it as slides, index the videos. So the interaction becomes much more. Now, I won't do this for Sanjay's presentation yet, or anybody else's presentation, because I want to think of it as a series first. So I'm doing it only for different series. But that's what we're going to do. All right, folks, thank you very much. And thank you very much, Sanjay. As always, great meetup.
Thank you. Thank you.
Take
Bye,
care,
everybody.
everyone.
Good night,
Thank you,
everyone.
bye-bye. Thank you.